everyone, and welcome to Chapter 12 Part 2a on respiration using other biomolecules. Now, in the previous two videos, we learned about aerobic respiration and anaerobic respiration separately. And it is both the process where organic molecules are broken down in a series of stages to release energy, which is used to synthesize ATP. This is an important definition of respiration, yeah? But so far, we have been talking about glucose non-stop. Actually, there are other molecules like amino acids, glycerol fatty acids that can be used in respiration as well and broken down to produce ATP. So let's start with what we know first. So we know that glucose is a respiratory substrate and is used in respiration. And actually, it is essential in neurons in the brain, red blood cells and lymphocytes, which solely use glucose as a respiratory substrate. So neurons in the brain require sugar, and that is probably why I'm hungry a lot when I'm working. Maybe, just maybe. No, I'm just kidding. Okay, anyways, glucose is oxidized in glycolysis, link reaction, and a Krebs cycle. Oxidative phosphorylation is not in this list because oxidative phosphorylation does not oxidize glucose. It takes the NDH and the FADH2 and turns it into ATP, right? Glucose is not oxidized there. Glucose is only oxidized in glycolysis, link reaction, and the Krebs cycle. Let's talk about fatty acids and glycerol, which are all in this group called lipids. Now, lipids are oxidized in slightly different manners. You don't need to know the details, but you need to know that it's converted into acetyl coenzyme A, which is a 2-carbon compound which can feed in directly into the Krebs cycle. So it skips glycolysis and link reaction, straight away converts to acetyl coenzyme A and then in a different process entirely, and then oxidize in the Krebs cycle. Number 3 is protein, so things like amino acids can be used in respiration as well. Okay, so how is it used? Amino acid is converted to pyruvate or acetyl coenzyme A, so it can participate in the link reaction or go straight into the Krebs cycle. So the amino acid, if it's converted to pyruvate first, then it will need to be oxidized in the link reaction and the Krebs cycle. But if protein or amino acid is converted into acetyl coenzyme A, then it will need to it will not need to go into the link reaction. It can go straight into the Krebs cycle. Now, in all these reactions, don't forget that NADH and FADH2 are produced and then passed on into the ETC in oxidative phosphorylation, and through that process produce ATP through chemiosmosis. But which molecule contains the most energy? So we know that there are 3 respiratory substrates. Which one, if I fully oxidize it, will give me the most energy? So let's work backwards here. Where does most ATP come from? Most ATP comes from oxidative phosphorylation, right? And what happens during oxidative phosphorylation? We are taking NAD and FAD, the reduced version, removing the hydrogens from them, and the hydrogen will split into hydrogen ions and electrons. Electrons will be passed through the ETC, hydrogen ions will be pumped into the intermembrane space, and it moves back to the matrix via ATP synthase, and this is down the proton gradient. And of course, the hydrogen ions will combine with oxygen to form water, but as it moves down into the matrix, the ATP synthase rotates and this produces ATP. And you can see here that a lot of the processes rely on hydrogen atoms and hydrogen ions and electrons. And therefore, we can safely conclude that the amount of hydrogen available would determine ATP production and chemiosmosis. So we can safely conclude also that the more number of carbon-hydrogen bonds you have per unit mass in a respiratory substrate, or the more hydrogen atoms in a respiratory substrate you have, then the more hydrogens can be carried by the NAD and FAD. Then the more reduced NAD can be produced. And if that is true, then there will be more oxidative phosphorylation, more hydrogens pumped across the inner mitochondrial membrane, and therefore there will be a steeper proton gradient, resulting in more ATP made per gram of respiratory substrate, and therefore the energy value of the respiratory substrate will be greater. So remember, the ones in red especially, the higher the number of carbon-hydrogen bonds per unit mass, the greater the energy value. Now at a glance, you can look at this hexose molecule right here and this triglyceride right here. You realize that, hey, sugar, carbohydrates have a lot of OH groups in them, a lot of hydroxyl groups. But if you look at lipids, there are a lot more carbon-hydrogen bonds per unit mass. And this answers the question, which molecule, what kind of respiratory substrate has the highest energy value? The winner is lipids, yay! So lipids has most carbon-hydrogen bonds per unit mass, or number of hydrogen atoms per unit mass. The per unit mass is quite important. Most compared to carbs and protein, so it wins. It releases the most energy per unit mass, and we say that it has a very high energy density. So since it has a lot more number of atoms per H atoms per unit mass, this means many more hydrogen is available to reduce oxygen to water, and we can expect a lot more water to be produced from the metabolism of lipid, because there's just so many hydrogens. So oxygen is a hydrogen acceptor, right, the final one, and therefore more water is produced as well. Now, it's important to note that lipids can only be oxidized in the presence of oxygen. Same for proteins, which we'll see in a moment. Okay, so proteins is in the middle between carbohydrates and lipids. It has slightly more carbon-hydrogen bonds per unit mass than carbohydrates, but still less than lipid. Just slightly more carbon-hydrogen bonds per unit mass. It's used usually during starvation or a lack of fat or carbs, so it's used at the last resort. Oxidation of amino acids also can only happen in the presence of oxygen, like we said just now. Okay, so here are the energy values of the respiratory substrates we know so far. These values have to be memorized. So carbohydrate has 15.8 kilojoules per gram. Lipid produces 39.4, which is a lot more than carbohydrate, right? Kilojoules per gram. And protein only a little bit more than carbohydrate, around 17 kilojoules per gram. You can see here that lipid has the most CH bonds and therefore has the highest energy value. Carbohydrate has the least CH bonds. It has more oxygen comparatively and therefore has a lower energy value. So you might be wondering, why in the world do we need to memorize these values and where in the world did they come from? Let me answer the first question first. Why do you need to memorize this? It's because exam will ask. They will show you the energy values and then ask you what respiratory substrate is used. So you need to know them. Anyways, the second question is, where did they come from? Well, they came from an experiment using this machine here called a calorimeter. Now, a calorimeter is a fancy name and this is a fancy setup, but essentially is very simple. Essentially, you are just burning a known mass of substance with oxygen and measure the rise in temperature of a known mass of water. Now, the idea is when you burn the substrate breaks down, and when it breaks down, it will release a certain amount of heat energy, right? The heat energy will cause the rise in temperature of the water surrounding it. And if you measure that change of temperature, you can figure out how much energy the respiratory substrate has released, in essence. So there is a formula to calculate this, but fortunately we do not need to remember it, and therefore you just need to know what this machine is called. That's pretty much it. So yeah, moving on from here. Let's talk about respiratory quotient. So just now we were talking about energy values, kilojoules per gram, but this doesn't have a unit. This is respiratory quotient, and basically is carbon dioxide over oxygen. Specifically, it is the ratio of volume of carbon dioxide produced to oxygen used per unit time. So ratio of the volume of carbon dioxide given out, breathe out, to the volume of oxygen used in a certain amount of time. And of course, you can use number of molecules or moles as well, as long as both on top and the bottom are the same. We can do this experimentally and measure the volume of carbon dioxide given out and taken in, or we can use equations to figure it out. Now, different respiratory substrate would have a different ratio, different respiratory quotient. Now, you might be thinking, I understand energy values. This means how much energy is that particular substrate giving out. But how about respiratory quotient? Now, the function of RQ is this, is for us to work backwards. If we can theoretically calculate how much RQ to expect, we can do it experimentally and figure out whether the organism is using aerobic or anaerobic respiration and what type of substrate is being used. So before we go into the experimental part, let's do some theory. So we're going to just do this for aerobic respiration of glucose, of fatty acids, and then anaerobic respiration of glucose. So in aerobic respiration of glucose, so in the presence of oxygen, we have this particular beautiful equation right here, right? So we can see 6 oxygen here is taken in, 6 carbon dioxide is given out. So RQ equals to CO2 over O2, right? So it's 6 over 6, which gives us a theoretical value of 1. Now, in aerobic respiration of fatty acids, we have this equation here. You don't need to memorize this equation, but I'm just telling you as an example. This is oleic acid from olive oil. And you can see here more oxygen is needed. Well, because it has a lot of hydrogens, right? And more oxygens are needed to be hydrogen and electron acceptors. So we have a lot more oxygen here compared to carbon dioxide. RQ is equivalent to CO2 over O2 again, so 18 over 25.5. So that gives us a value of 0.7. Now, this RQ level is different from the aerobic respiration of glucose. It is slightly lower. So keep that in the back of your head first. Let's do a few more theoretical values. In the anaerobic respiration of glucose in yeast, so the ethanol pathway, we have this equation, right? Glucose is used, and that's converted into ethanol and carbon dioxide. There's no oxygen being used here, but there's 2 carbon dioxide being produced. The RQ that we expect would be 2 over 0, which is infinity. Now, in reality, this is the theoretical value, right? But in reality, when we do the experiment, we are not going to get an infinite value. We will expect some cells to have aerobic respiration and some cells to not. And therefore, in like the beaker with a lot of yeast cells, that value won't reach, the RQ value won't reach infinity. It will be just very high. So we just consider high values of RQ as anaerobic respiration is occurring. So not necessarily infinity, just a very, very big number. But this is in yeast, right? How about in animals? How about in muscle cells, in the lactate pathway? So in the lactate pathway, it's different because no oxygen is used and no carbon dioxide is used as well. So we expect a RQ value of 0, isn't it? 0 over 0. So no RQ can be calculated. So we can see here in the summary table, what we can expect. Carbohydrate would be 1, like glucose, 6/6. Lipid would be around 0.7. Protein is a little bit higher than that. We don't need to know why, we just need to know that it is a little bit higher. And anaerobic respiration in yeast would be greater than 1 or approaching to infinity, so a very huge value. Anaerobic respiration in muscles would be 0, so we cannot calculate the respiratory quotient anyways. These values again need to be memorized for each respiratory substrate as well as anaerobic respiration. So we have learned how to calculate the theoretical value of respiratory quotient, RQ. But now we need to learn how to measure and do an experiment to find out the experimental value of RQ. And to do that, we will need this setup here called a respirometer. Now this setup is pretty comprehensive, and don't worry about it, we'll walk you through it. But basically the general idea is to measure the rate of oxygen uptake during respiration by specifically small terrestrial invertebrates or germinating seeds. Why does it have to be small terrestrial invertebrates? Well, because we need to fit it in a test tube and doesn't have water, so it has to be invertebrates that survive on land. So it could be a grasshoppers, it could be cockroaches, okay, it could be some mealworms or things like that. Or we can use germinating seeds. Germinating seeds because they don't undergo photosynthesis just yet, they undergo respiration only. So they have not have leaves yet, so they can't photosynthesize. So how does this work? So let's look at simpler sort of setup here. The idea is you need 2 vessels connected by this capillary U-tube that has some fluid inside. We call this a manometer because it senses pressure changes. Or it can just measure the volume of gas in both these vessels. So at first we need to keep it at the same level, and later on it will move. Okay, so what is important is that throughout the experiment, we keep the pressure and temperature constant, especially when the reading is taken. We can use a thermostatically controlled water bath here to make sure that it's a reliable experiment. We have to use a control tube. So the reason why we have 2 vessels is that one has a specimen and the other has an object of equal volume of or mass compared to the specimen and the absorbent. Okay, and this is to compensate for changes in the atmospheric pressure. So this just gives us some control over what's going on there. Number 3 is the most important thing of all. The most important thing is to have soda lime. Now soda lime, or maybe potassium or sodium hydroxide, will absorb CO2. And this is very, very important because only when we absorb the CO2, then we can measure the rate of oxygen consumed. Okay, when the organism breathes in, the oxygen taken in will affect the manometer scale. Okay, and it will move up here nearer to the experimental tube. So level will increase here because of oxygen consumed. But when the specimen breathes out and the carbon dioxide is given out, is going to be absorbed by the soda lime, and therefore it will not affect the level of the manometer tube. So you can see the volume of fluid here, you can measure the volume, okay, calculate it a little bit, and you'll be able to find out the volume of oxygen consumed. Now if you think of RQ though, we need both values. We need the the volume of oxygen consumed as well as the volume of carbon dioxide produced, right? CO2 over O2 basically. So how do we do that? So we have to do the experiment twice. We use the same apparatus, same organism, same temperature twice. So at first we will use the setup with soda lime that I just described to find the volume of oxygen taken in. But then in the next experiment, we are going to remove the soda lime, remove the chemical that absorbs carbon dioxide, and we repeat it again so that the manometer scale will be affected by both oxygen consumption and carbon dioxide release. But because from the first experiment, we already know how much oxygen is consumed, so with that knowledge, we can calculate experimentally how much carbon dioxide is released. Okay, so we do it twice, one with soda lime and one without soda lime. In one, we are looking for the volume of oxygen consumed. In the next one, we use the knowledge of the first experiment, in order to find out how much carbon dioxide is released by the organism. So here are some examples. So at first, we would have soda lime like this, right? This is the beginning of the experiment. With soda lime means that the carbon dioxide is absorbed. The level will go up because oxygen is absorbed, is consumed, it's taken in by the organism, right? If oxygen absorbed equals to carbon dioxide produced, okay, you expect that when you don't have soda lime, when it breathes in, it's going to take in oxygen, the same amount that you did just now, but when it expires, when it breathes out, oxygen will be produced, so it will lower down the level of the manometer scale. So if it looks like it does not change here, okay, the final result, it looks like it's still at the same line, this means that oxygen absorbed equals to carbon dioxide produced, and the respiratory quotient is 1. So this means that aerobic respiration of glucose is going on. This is the second scenario. This scenario, carbon dioxide produced is more than oxygen absorbed. So what would we expect in this case? So at first, this is the beginning of experiment, right? With soda lime, it's going to increase because this fluid here displacing the volume of air is showing you the oxygen being absorbed. So it moves up because the oxygen is being absorbed by the organism, whereas the carbon dioxide produced is absorbed by the soda lime, so it doesn't count here, doesn't see here. But without soda lime, okay, this volume is X. Without soda lime, imagine the organism takes in the oxygen. We know that it's probably this much oxygen that takes in, and when it breathes out a little bit more. So more carbon dioxide is produced. So you expect it to go below the line there. So even more carbon dioxide is produced than oxygen absorbed. Now we look at this value here, Y. Okay, we always compare it to this line. Look at this value Y, and we can estimate that the number of carbon dioxide produced, the volume of carbon dioxide produced, is probably X plus Y. Whoops, X plus Y. And oxygen is X, so it's X plus Y over X. So you get an RQ that is more than 1. And if it's more than 1, you know that in this case, it is an anaerobic respiration that's occurring. Okay, what if the oxygen absorbed is more than the carbon dioxide produced? What kind of results will we see? So if you get the drift already, pause this video and try to imagine it first, or try to draw it out first before you see this to confirm. Okay, pause it and then try and play it again later. Okay, here we go. So this is the initial stage of the experiment, it's the same. Now, with soda lime, the oxygen absorbed is going to be measured. So the volume here is going to increase because you're sucking it up. So it's sucked up a little bit. So it's high. So this is called X. This is X cm cubed of volume of oxygen being absorbed. It's with soda lime. So here, again, you don't see the CO2 having any effect on the manometer scale. But without soda lime, we know that, hey, this is what happens when the organism breathes in. So this is as the volume that it breathes in. And when it breathes out, you realize that it drops a little bit. So it only drops here. It doesn't drop all the way, it just drops here. So it's a little bit higher than the original blue color line here. So this one is saying that if 1 meter fluid increases by Z-cm, this is what we're calling this, we know that oxygen absorbed is more than the carbon dioxide produced. And the RQ level is X minus Z. Okay, X minus Z is the carbon dioxide over X, which is the oxygen consumed. And therefore, you will have an RQ value that is less than 1. So in this case, your organism is probably using lipid or probably using protein as a respiratory substrate. So that's using the respirometer to measure RQ. But hey, the respirometer actually has many more applications. So not only to measure RQ, you can use the respirometer. Okay, don't remove the soda line, put the soda line inside, and measure the effect of temperature on oxygen consumption. So you do the same thing, you have the same setup, but with the soda lime definitely in there, and you take the measurements of respiration, of volume of oxygen consumed at various temperatures, and you can plot the graph of oxygen consumed against temperature. The expected result should be this: the higher temperature, the higher rate of respiration, and therefore the faster the absorption of oxygen. However, this has a peak and reduces after a certain point, because after that enzymes start denaturing and the processes slow down. So that is the respirometer. Now for the last part of this particular video, I want to talk about redox indicators. So far, we have learned how to measure the rate of respiration of small invertebrates and of germinating seeds. How about yeast? So we can use these redox indicators to investigate the rate of respiration in yeast. So the redox indicator actually does not damage a cell, which is quite useful. So we can add it directly to the suspension of yeast cells, usually in a test tube like this. And when it's reduced, when respiration occurs, the blue dye will become colorless. So to measure the rate of respiration of yeast, all we need to do is to measure the time taken for dye to change from blue to colorless. So this is not measuring oxygen or carbon dioxide, it's just measuring the rate of respiration in general. So if you measure time taken for dye change to blue to colorless, this would equal the rate of respiration of yeast. And of course, in paper 5, they'll ask you to describe an experiment, and you can talk about different independent variables you can use. So you can do this with different temperature, you can use different concentration of substrates, or different substrates even, give the yeast different things, and then measure the rate of respiration of yeast and plot a graph. And therefore come to a conclusion about what its effect on respiration is. So that's it for me now. Don't worry about the experiments part. I think the more you do paper 5, or the more you do questions related to this, then the more you get a clearer idea of what's going on. I will come up with a video to teach you how to answer paper 5 questions on planning experiments as well. Now, for all experiments in all my slides for A2, there is a little paper 5 sort of box, yellow box at the right hand corner, and therefore if you're revising for paper 5 in the future, you can refer to this yellow box in order to memorize or familiarize yourself with some of the processes. So that's it for me today. See you next video. Bye!